I'm here in Bedford, Massachusetts, in a wonderful garden talking to Professor Ernst Mayr, who has seen more of this century than anybody I can think of, and anybody who's likely to be in the audience, and probably any biologist. And I'm going to ask him about his life and what he recollects about this century, and also what he thinks about the future of biology.
Well, it is perhaps not an accident that I've become an evolutionist. I was a very fortunate youngster because both of my parents were intensely interested in nature. And every weekend they took me and my two brothers, one older and one younger, on excursions. And one weekend we went to a heron colony, another weekend we went to a quarry where there were lots of ammonites and other fossils. My mother was very good in identifying mushrooms, not only the edible and the poisonous ones, but she knew just about every mushroom. In the early spring we would go to the vineyards in Franconia looking for rare spring flowers and so on and so forth. And of course I soon learned birds. I knew every voice of every local native bird, but I was actually not going to become a biologist. I was to become a medical person, a physician, because that was the family tradition. My father's brother was a doctor, my grandfather was a doctor, his father was a doctor, his father was a physician, and so I went off to university to become a medical man. However, just before I went there, I saw a rare bird, at that time extremely rare. It was a red-crested pochard, the first record by anybody made in 1923 since 1846. And to make a long story short, through that I met Stresemann, the ornithologist at the Berlin Museum, and he in due time persuaded me to shift over to biology, and then he would be able to arrange it that I could go on an expedition. And so it happened, and finally Lord Rothschild engaged me to go to New Guinea and search on 3 different mountain ranges for certain rare birds of paradise.
That sounds like a great adventure for a young biologist. How old were you then?
I was 23 when I went out there. And I was definitely a greenhorn. When I was being recommended to Lord Rothschild as being his collector, it was not mentioned that I had never shot a bird, that I had never skinned a bird, and all that sort of thing. And I got a very quick course in all this, and furthermore, and that saved my life, when I passed through Java, the Dutch Colonial Museum in Java, in Buitenzorg as it was called in those days, now Bogor, lent me 3 experienced expedition assistants, so-called mantries, who were highly experienced in all these things. And while I was the leader of the expedition in New Guinea, they taught me all these tricks that you have to know. These mountain ranges, 2 of them had never been visited, 1 only incompletely so. Well, subsequently, I immediately, after the New Guinea work, I joined an American expedition which on a schooner visited many islands in the Solomons, and I came back after 2.5 years as a truly experienced naturalist. Well, I never went back into medicine. I got a position first at the Berlin Museum, then at the American Museum, and finally in 1953 I was offered a professorship at Harvard.
What made you into an evolutionary biologist? You were an ornithologist, but what made you take that next step?
Well, as soon as I learned a little more about evolution and the status of evolutionary biology, I realized that we naturalists had entirely different ideas about evolution than the geneticists. The geneticists at that time thought genes did everything, and the earliest post-Mendelian geneticists, people like Bateson, De Vries, and Johansen, they thought a single mutation would make a new species. Now, we naturalists knew that speciation was a very gradual process, so we look for a gradual explanation of speciation and evolution, and the only one that was available at the time was Lamarck's explanation, that by gradual adaptation and inheritance of acquired characters and so on and so forth, eventually a new species would develop. Well, by the time the 1930s came, there were two clear-cut camps. The one was that of the geneticists, led by people like R.A. Fisher, J.B.S. Haldane, Cyril Wright, H.J. Muller, and up to a point still, T.H. Morgan, and they were very reductionist. Everything was brought down to the level of the gene. However, they had clearly understood the gist of the Mendelian message, which is that the genetic material is constant, it cannot be changed by the environment. The Lamarckian thesis is absolutely refuted, and also that the genetic material comes in small little packages called genes, and the genes of mother and father do not blend and mix and fuse in the offspring, but remain discrete and may again recombine in the next generation. Well, this message of the geneticists that developed in the first 30 years of the century had not yet reached really the naturalists. Neither had their finding that the diversity of nature was not types, and that what is exposed to natural selection are not genes, but whole organisms, whole individuals. Furthermore, the naturalists, as I said, they knew that evolution is very gradual. They knew that speciation happened by certain populations being isolated for a long time and acquire during that isolation certain genetic characteristics which permit them to coexist with their parental populations and, in other words, being different species. Well, here we had two camps, both with some very important information, but not knowing the information of the other camp.
What changed all that? What brought the two camps together?
A Russian geneticist now living in America, Feodosit Stebzhansky, who in 1937 published a book, Genetics on the Origin of Species, in which he used his own experience as a young naturalist in Russia, where he became a beetle specialist and really understood species and speciation, and 10 years of work in T.H. Morgan's laboratory in America, where he became an outstanding geneticist. Combining both of these knowledges, he was able to bring these two camps together because he was able to present a story of evolution which was equally compatible with the thinking of the geneticists and that of the naturalists.
What was the next step?
I made my contribution, and this all happened in the 10 years after Stebzhansky published his book in 1937, where a whole series of books was published by people like Huxley, myself, George Gaylord Simpson, the paleontologist, the German zoologist Bernhard Rensch, and the botanist Ledyard Stebbins. And my own contribution in this was primarily to clarify the species concept, and my writings led to the general adoption of the so-called biological species concept, which considers the species a reproductive community rather than a batch of different individuals. And the other step was my contributions to the study of geographic speciation and showing the pathway by which speciation happens. Now, there was not a complete understanding yet of the two camps in one respect. For the sake of simplicity, the geneticists still assumed that the gene was the object of selection, the unit of selection. While we naturalists, we knew that this was not the case. A gene is never isolated from the other genes. It combines with the other genes to form a genotype and produce a phenotype, and it was this phenotype which was really the target of selection. And it took almost 40 years before, in the 1970s, the geneticists finally gave up the idea that the gene was the target of selection. Dawkins was one of the last to believe that obsolete idea, and from the 1980s on, everybody agreed that the individual as a whole was the primary object of selection. However, I, in some rather recent writings, point out that it wasn't the only object of selection. When we deal with social organisms that live in small social groups, quite often the social group as a whole is the target of selection, because the interaction and cooperation of the individuals, of this small group of individuals, is what adds fitness to the whole group as such, a fitness beyond the added up fitness of the individuals, and that then the social group as a whole is the target of selection. And that idea, which I've promoted quite strongly recently, explains one of the great riddles of evolution. In Darwin's day, and up to, as I said, among the geneticists, right up until the 1970s, with the individual always being considered the only target of selection, it was very difficult to explain altruism, the evolution of altruism. How can, if only personal advantage is of selective value, then how can altruism develop, is the question. But the minute you admit, no, it isn't just the individual, the social group as a whole can be a target of selection, and in a social group, cooperation, altruism, helpfulness, all these features, all these attitudes that are altruistic, is of tremendous selective importance, because it adds to the selective value of the group as a whole. And so here is an advance in evolutionary thinking that has happened in the last 15, 20 years that has really added a whole new dimension to our thinking, and it has made a great riddle of evolution, namely how can this egotistical process of selection produce altruism. It has finally solved that problem.
What do you think are the most important areas of evolutionary biology right now and in the future?
To begin with, it seems to me, and I know I'm probably a minority now, that the basic theoretical framework of evolutionary theory, as originally established by Darwin and as filled out later on during the so-called evolutionary synthesis of the 1940s, that basic framework is extraordinarily robust. I think it has been able in the last 50 years to refute every new theory that has come up and shown it to be invalid, and has reconfirmed and re-established and really re-strengthened what had been the belief of the leading evolutionists all along during that period. So I think as far as the basic theory is concerned, we are rather safe, except for one thing. Almost all of our thinking about evolution is based on birds, butterflies, and a certain series of what you call model organisms. For instance, it now has turned out that, of course, bacteria, which are organisms too, and the first 2 billion years of life on Earth was all bacteria, that bacteria have no sexual reproduction, they get their genetic variation by simply unilateral gene transfer. And what is transferred and how it is incorporated and all that is still very vague, because even the two most distinct group of bacteria, the so-called eubacteria and the archaebacteria, exchange genes unilaterally. And to make a joke, since they are thereby a reproductive community, by definition, the eubacteria and the archaebacteria are one biological species, you see. Now that shows you the absurdity of this kind of thinking. So here we don't really know what happens at that level. Yeah, the eukaryotes, how they were formed, how that nucleus originated, how they incorporated other prokaryotes as cellular components, how all this works together, how they started with sexual reproduction, how they developed mitosis or meiosis, all these sort of processes have to be reconstructed.
What are your thoughts on cladistics and classification? It's a tricky subject.
Well, I have right now completed, but not yet published, a very large paper entitled On Classifications and Other Ordering Systems.
Marvelous.
Now let me go back. This all starts with the year 1859, when Darwin had a whole chapter in The Origin, chapter 13, in which he said how one should classify. And he clearly proposed classification has to be based on two criteria. One, similarity, and secondly, and that was a brand new idea, you couldn't have a grouping of the descendants of common ancestors, and that is what all the groupings of organisms are, without taking that factor into consideration. And he said, when you delimit a taxon, a higher grouping of species, that grouping has to consist only of the descendants of the nearest common ancestor. In a real classification, you group species together and then, let's say, put them into a genus, then you take related genera and group them together into a family, and you get a beautiful hierarchy, and you get that whenever you do classifications, wherever you go, are always based on similarity and can be thereby hierarchically organized. Well, if you go to a cladistic arrangement, you get a group which one German author recently called mammalia, mammals, and it started with pelygosaurs, some very primitive synapsid reptiles, and then went on to the therapsids, and finally there was the change in the jaw and all sorts of other things, and finally out came the mammals. And all that he puts on the mammalia, because that what is forced by his cladistic principles. Cladistic classification, I call it cladification, which is the right word because it's not a classification. A cladist doesn't make classes that these things, it makes clades. Cladification is simply not a classification. It may be useful for some phylogenetic purposes, but it is not a classification, and there's no argument that can change that assertion.
How do you assess the contribution of molecular techniques to classification?
You have to remember that there is one method, one methodology, which Hennig favored and promoted, which is completely sound, and which I use, and which all good classification people use. That is the so-called cladistic analysis. And that is, if you're uncertain as to how you see, Darwin said, everything has to be derived from the nearest common ancestor. Well, how do you find that nearest common ancestor? And that is where you use the method of cladistic analysis. And the crucial thing that Hennig proposed, and he himself says that it's not new with him, other people had done it before, is to base his branching pattern, and that's all that's involved here, entirely on derived characters. never use an ancestral character, so what he calls a plesiomorph character. Use only derived characters, and then you get, on the whole, there are difficulties, but they are inevitable. You get a pretty good branching pattern. And so what now happens is, the cases that you are familiar with in mammals, they looked at subjected the insectivora and an order of mammals to a cladistic analysis, and they found that 2 of the families included in insectivora, the golden moles of Africa, South Africa, and the tenrecs of Madagascar, absolutely didn't fit. So naturally, they have to be taken out, and every Darwinian classifier totally goes along with that. But that's only the cladistic analysis to determine something that Darwin had insisted on, the descent from the common ancestor. And of course, you have the same with the hippopotamus, which is now shown to be most closely related to the terrestrial ancestors of the whales, and so on and so forth.
What field of evolutionary biology is growing now and will grow in the future?
Well, I think this exciting new period has already started because of the recent developments in developmental biology. When the evolutionary synthesis happened in the 1940s, the developmental biologists curiously refrained from participating. In fact, they were openly hostile to the synthesis. They were openly hostile to natural selection, and they simply said there was no connection between what they were finding in development and the evolutionary phenomena. But now it is quite clear that since the phenotype, which after all is where evolution happens, is being made by the genotype, and that the development is the pathway from the genotype to the phenotype, and furthermore, that the changes in the genotype are influenced by development, we now realize that the study of development is indeed a very important part of evolutionary biology, and we are very happy, even though it is part of functional biology, it is intimately connected with evolutionary biology, and we are very glad that the developmental biologists have now taken up this work. What is interesting is that they had to go right down, not only just to the level of genes, but to the level of molecules, to the base pairs, and to the details of the genes in order to make sense of what is going on in development during evolution. Well, this field is it's less than 20 years old, and we are only at the very beginning, and this is where one of the major frontiers of biology is.
You know, I'm really interested in brain function and other complex systems, and I know you've thought a lot about complex systems. Would you like to say something about it?
No, it is that business of taking a complex system like the brain, or like a fully formed organism, or like an ecosystem, and studying all the details by analysis, a very legitimate procedure, and then trying backward, upward again, putting things together in order to fully understand the system. And as soon as we do that, we get into the business of the interaction of these units. And it is the interaction that is the thing that is hard to study, hard to explore, hard to determine, and yet it is these interactions that are the crucial thing about the working of systems. And this begins at the very lowest level. It goes back to the time of Darwin's friend T.H. Huxley. He was asked whether one could determine what water is by taking it apart. And he said, no, you cannot. You take it apart and you get one gas, and it's called oxygen, and you get another gas, that's hydrogen. And if you have these two gases, you don't have water. You still don't know what water is. It's the interaction between oxygen and hydrogen, and very particular interactions that make for water. And when some new property emerges, like the liquidness of water, or as Huxley called it, the aquosity of water, when that emerges, when you put things together, that is called emergence. And this is one of the big battles in philosophy of science, and in evolution, and in biology in general, and in particular, that emergence is an extremely common phenomenon. You find it in any complex system, and yet to study emergence and determine what makes the new characteristics emerge, those studies are, for the most part, very much at the beginning. Yes, there has been a philosophy of science, and there is a philosophy of science, and a science of science, but it deals only with some very basic principles and phenomena, like science has to be objective, it does not accept supernatural forces, and so on.
You know, I'm really intrigued. My own view of biology is that it's a source of wonder and joy. What parts of your life have given you a sense of wonder and joy?
Well, joy and wonder, if you use this, actually came not from purely intellectual enterprises as my research later on, but I would say that probably my years in the wilderness of New Guinea with very lovely people, and subsequently 9 months of sailing through the South Seas and the Solomon Islands, that was an experience, or a set of experiences, that cannot be surpassed. And even though they were combined with an enormous amount of hardship, I had every possible tropical disease, and I sometimes almost starved to death. I sometimes got shipwrecked. I had all these difficulties, but they all absolutely blanch. They disappear in comparison to the memory of all these wonders of nature that I saw, and mankind with the natives of New Guinea that I experienced in those years.
Thank you very, very much for sharing all this with us. It's been a long interview, but we really appreciate it.
Oh, you're most welcome.
Joy and wonder to a lot of people, you know.
So now I'm going to get out of this sun because I'm sizzling.